Good morning, all. Excavata. So you may recall from our major clade presentations that excavata are one of the major eukaryotic clades. Here they are. And they're one of the more uncertain. So they're drawn here with a dashed line. Both their monophyly, whether they're a real group or not, and their placement on the tree is uncertain. So here's just a blow up of these. I'm not going to cover all, just a couple of these groups here. I do want to point out the euglenids are the only photosynthetic lineage, and I'll say just a few words about them. The putative synapomorphy for euglenids is this excavate feeding groove that was presented in major clades, and we'll talk briefly about with our readings. Among the more well-known and major clades of excavata are euglenozoa and heteroloboscia, and I think I'll focus all of my remarks on these two. There are other lineages in like Giardia and Trichomonas, which we talked about because they lack mitochondria. But for today, I'm going to focus on these really diverse lineages. Euglenozoa consists of two major clades, the euglenids, including Euglena gracilis. These are friendlies, and they include the photosynthetic lineages, and the kinetoplastids, most of which are friendly, but are most famous for us for trypanosomes, for African sleeping sickness, and leishmania, which can be a flesh-eating disease. So euglenids tend to have two flagella, as do most eukaryotes, but they have this weird complex pellicle, which I'll show you some pictures of, that give them very strange movements. About only a third of euglenids have plastids, and 2/3 are heterotrophs or saprotrophs. They eat dead things. And even these plastid ones, many of them, if you put them in the dark, they can transition back to heterotrophy, which suggests a recent acquisition of plastids. Here's just a picture of some of these euglenids. We actually saw Facus the first day when we did our demonstration lab all together. And here they are again in a scanning electron micrograph. And here you can see these crazy protein folds on the outside. These are this is their pellicle. Here's a cross section now through their pellicle, this very rich protein coat on the outside of euglenids, and it's this pellicle that gives them their really distinctive movement. So we'll just take a quick look at one of these guys in a light microscope. You may be able to see two flagella, and it moves by this process called metaboly, which is underlaid by that complex protein coat. So I think we'll watch this for about 2 minutes. Again, people have a lot of time to make videos. Okay, I think we can end with the psychedelic whales in a Black Sea. I suppose that is a valid analogy for what they look like. So the euglenids include the photosynthetic lineages, and they have this strange protein coats in the metaboly. The kinetoplastids, as with most microbes, most are friendly or don't harm us. They have two flagella. They have this strange undulating membrane, and I already told you about their maxi mini circles in their mitochondria. So here's a classic drawing of a kinetoplastid. They have these flagellas. Sometimes they only have a single flagellum, but this one has a smaller one and a long one with a membrane. So this gives them a really distinct appearance, the membrane on their flagellum. Here's dividing cells. And if you look at a cross section, each of these is a microtubule, and here's a eukaryotic flagella. It would be nice for the exam, and I will put something else up about this before the exam, if you knew that all eukaryotic flagella, with few exceptions, have this 9x2+2 structure, 9 pairs of microtubules and 2 in the middle. I will do a mini lecture on that and post it. Anyway, these are microtubules, these are microtubules, this is the flagellum. And that was a cross section through one of these cells. Here's the undulating membrane. Here's some red blood cells with a trypanosome swimming through. And here's their strange mitochondria with this kinetoplast DNA, the maxi and mini circles. So if you may recall, I explained that the maxi circles have broken genes that are corrected by RNA editing using mini circles. So here was an example of an edited gene. The black is in the DNA, and the red and blue are alterations made in the RNA in RNA editing. And RNA editing is widespread, but this crazy maxi-mini circle is unique to the kinetoplastids. So it starts with this black bit in the maxi circle, and the mini circle comes in and corrects, which allows another mini circle to correct. And you either have the addition of U's or the deletion of nucleotides to give rise to your functional proteins. An example of a friendly or non-harmful kinetoplastid are bodonids. We see these all the time in lab. They're teeny little heterotrophic flagellates, 10 microns, about 10 E. coli long. They're free-living, they can be endosymbiotic, they can be commensal. I guess there are a few that are parasitic on non-human animals. But we know most about the trypanomastids, including trypanosoma and leishmania. These are two general. They're parasites. I'll say a little bit about their life cycle, and then I'll talk a little bit about their variable surface glycoproteins that allow them to escape the immune system. Here's a colorized image of red blood cells with the trypanosomes sitting on top. So African trypanosomes can cause trypanomyosis or African sleeping sickness. It's a sometimes fatal neurological disease. It's found in sub-Saharan, widespread throughout sub-Saharan Africa, transmitted by the tsetse fly, and it remains a really huge problem, in part because of the problems associated with healthcare in socioeconomics and war and poverty. So sadly, not unfamiliar story where healthcare is intersects with economics and governments and nationalism. So here's just the life cycle of Trypanosoma brucei in the mammal. It takes on these forms. They have much less exciting names than in the apicomplexans we saw before. When they're bitten by the tsetse fly, they go through development in the midgut, then they actually swim to the salivary glands of the tsetse fly and wait there to be injected into an animal. And to give you a sense of that, here's a tsetse fly with a blood meal in its belly. So much of the development goes on here, but when they're developed, they swim up to the proboscis. So here's the life cycle. They come into the gut. They can also develop in here and enter the salivary gland. And if you looked in a salivary gland, one of these infected tsetse flies, you would see it full of trypanomastids. All of these are trypanosomes waiting to be ejected. Here's the little proboscis, and here are trypanosomes coming out of this tsetse fly. So this is a very high population sizes. One of the things that the trypanosomes do that's quite crafty is they have these variable surface glycoproteins that are underlaid by developmentally regulated chromosomal rearrangement. So we talked about crazy genome features earlier in the semester, and here's another one where they basically rearrange these proteins and only activate one or in the cell at a time. So in the end, here's the cell membrane, and they put these proteins on top. But what's crafty about them is they're constantly changing these proteins as if somebody stole your knapsack, and as they ran away from you, they changed their appearance, their clothing, their hair color, their height, etc. So these are the proteins on top of the membrane. Here's these microtubules and the cell membrane, and this is all of these thick proteins. These, there's about 300 to 1,000 variable surface glycoprotein genes in trypanosomes, so 5 to 10% of the trypanosome genome is devoted to escaping immune systems. One of the reasons why they're really hard to treat. I'll end with a friendlier group, and plus a negative in here too, the heterolobosea. Many are amoeboflagellates. They can be an amoeba 1 minute and a flagellate the next. So here's the nasty, Naegleria fowleri. It's thankfully very rare, but if you get it, you're likely to get a fatal meningitis or encephalitis. And you can get this by jumping into contaminated and usually stagnant water. So it has to be pushed over the blood-brain barrier to get into the brain. People also can get it from neti pots that aren't well cleaned, and it's a very serious disease. This organism can be an amoeba or a flagellate. So here it is as an amoeba and a flagellate, and this transition takes 20 to 30 minutes. The life cycle is such that it starts off as a cyst, it can turn into this amoeba, and then transition into a flagellate. So amoeba, flagellate, I guess it's saying here, this species can take 2 hours. So we'll watch this in a quick video. Here it is as an amoeba. It has these little pseudopods sticking out of it, not flagella. Then it changes its mind and it sits down, and this is sped up some number of times, I'm not sure, but after a period of 30, 60 minutes, it is changed from its amoeboid phase into the stage with flagella. So I don't think that was the best video we've ever seen, but I hope you get the idea that this organism can take on these different life history stages. Naegleria fowleri gets in through the nose, or you can breathe in, but the big one is through your nose. It can cross your blood-brain barrier, and it can cause these huge lesions in your brain, and it is, unfortunately, at this moment, not treatable. So to end on somebody friendly, the acrasids are one of the kinds of cellular slime molds. You find them on dead plant parts, tree barks, dung, soil, out there in the forest. Excuse me, acrasids can have flagellate members and non-flagellate members, and here's their life cycle. So you can start with a spore, it releases a little amoeba, the amoeba divides and divides and divides, and eventually they aggregate and they form these very complex, beautiful fruiting bodies. We're going to see this more in our multicellularity meeting. Here's a video of one of these acrasid amoeba. It has just these weird pseudopods on it, but it's out there eating bacteria. And when the bacteria run out, the cells aggregate. That's the observation. The hypothesis is that they do this aggregation as a means of increasing their dispersal potential. So here is a video after they have aggregated, and they form teeny little multicellular fruiting bodies that can emerge from the soil and are argued to be a dispersal mechanism. So that's all I have for the Excavata, which is this group in here. I look forward to seeing you all again.